Thank you very much, everybody. And here is my pleasure to introduce Professor Vadim Gladyshev. He's a professor of medicine in the Dana-Farber Harvard Cancer Center, and he's going to talk about quantifying aging and rejuvenation. So thank you very much, and the stage is yours.
Hello, do you see my slides?
Yes, they look great.
Yes, we can see.
Yeah, okay. So, well, this is a conference on aging and epigenetics. So because of aging, we need to ask the question, what is aging? And apparently, there are different views in the field. Some people think of aging as increased chance of dying with age. Some people think of age-related changes or decreased fitness or functional decline. Some people think of continuation of development or increased biological age or damage accumulation. Apparently, there are differences in opinion on the very fundamental question that we study. So our kind of goal is to try to understand that and we utilize genetic approaches for that. So in particular, the way our lab works is that we study longevity. So we study longevity across cell types, across mammals, across interventions, and these are primarily omics-based approaches, which result in what we call signatures of longevity. These are omics patterns that indicate a potential to live long. At the same time, we study aging, and here we develop omics also based patterns that indicate the transition through life course. And here we develop epigenetic clocks, but also transcriptomic clocks and other biomarkers. This lead to actionable biomarkers of aging. So, and the third component of our work is that we attempt to study the kind of reversal of the aging process, where we studied the transition from the older state to a younger state. This would be rejuvenation. And altogether, these three components allow us to target aging and rejuvenation and develop candidate interventions, whether they are nutritional, pharmacological, or genetic, and other. So it's a kind of overall view, and I will focus today more on this side, on the aging side, and which is primarily epigenetic studies, but not only. So as of course already, I mean, everybody knows, though, there's a huge development in the field, is the development of aging clocks. And this was started by Steve Horvath already more than 10 years ago. And now there are many very famous clocks like his clock, but also clocks, Hanumatol clock developed by Morgan Levine and Ake Lu, also with Steve Danbelsky, and many other clocks on the human side. We contributed on the mouse side. These are some of the clocks. In blue are the clocks developed in our lab, but also there are other clocks from other labs. And it's a very active area of research, as well as other clocks, not just epigenetic clocks. So just as an example, this is the first clock that we developed already 6 years ago. This is the mouse, actual age, predicted age. You see there's a very good correspondence in this mice that were subjected to calorie restriction for various periods of time, and you can see that based on the clock, they are younger. The same thing is for when we compare chronologically identical wild type and growth hormone receptor knockout mice. The knockout mice are younger. The clocks also work when we study reprogramming of fibroblasts to iPS cells. So just a couple of examples. Like within the last year, we published a couple of studies, and in this particular study, we addressed the question of whether mortality like increased chance of dying with age could be captured, or kind of differences in mortality changes with age could be captured by epigenetic clocks. And in this particular model is the NACMO rat, which is the animal that sometimes is discussed as non-aging animal. And the reason is that various studies, here is Sherry Buffenstein's data, show that the chance of dying does not increase with age. Then the question, of course, whether this kind of demographically non-aging animal actually ages or not. So does it age? So we be able to develop an epigenetic clock for this animal. These are animals of different ages, and you can see there is a clock, very good clock can be built, and therefore we think that it ages. So this is a paper. Also, there is another paper with Vera Grabunova and Steve Horvath showed the same thing basically. And here we compare aging rates of the NACMO rat, mouse, and human. And you can see, for example, for let's say decreasing methylation size, this is mouse, this is NACMO rat, this is human. Clearly, NACMO rat ages faster than human, but slower than the mouse. So I think this study raises a big question on the nature of aging. So of what, not nature, but what we call aging. Because if the chance of dying does not increase with age, but we can clearly see aging at the epigenetic level, the question is, does this animal age? And we think it does. But again, so further studies are needed, and because there remains to be a difference in opinion. Another study that we done recently is that we studied, or we tested the hypothesis that the biological age is does not steadily increases like chronological age, but kind of fluctuates. Sometimes it may be a little bit higher, a little bit lower. Of course, overall it increases, but it may be not a constant. Maybe adjusted on a short time scale. And here we studied the effect of stress. We found that severe stress increases predicted biological age based on epigenetic clocks. And this is both in mouse and human. And then once the stress is relieved, then actually age goes down. And we found this in the case of a major surgery, for example, like a trauma, like hip replacement and other surgeries. Also in the case of COVID-19, when subjects first go to the office, to the hospital, then to ICU, to the ventilator, and at that point the age is highest, and then once they recover, age goes down. Another example is pregnancy. So we found that the predicted age is highest in the third trimester in human, also in mice, and then postpartum it decreases. And also we describe the effect of parabiosis when young mouse, upon connection to the old mouse, temporarily increases predicted biological age, but following separation, it also goes down again. So we do not call it necessarily rejuvenation. So just more studies are needed, whether it's a true rejuvenation or just kind of reversible changes in biological age. But we think that it's not just kind of artifact, but actually changes in biological age of mammals that can be captured by epigenetic clocks. So another example is rejuvenation, and here we study again parabiosis, where we connect old mice and young mice for 3 months, and then after 3 months we separate them. This is a collaboration with Jim White at Duke. Interestingly, what we find that old mice, when they are connected to the young mice for 3 months, they live longer, and than control mice. So of course we start with old mice, and then so we're able to extend their lifespan. This can be captured at the level of epigenetic clocks. Here you can see we compare these mice upon separation, and also after detachment, the effect is still there. this is in the liver, also in the blood. So the effect is very robust. And this is just some of the clocks. These are clocks based on our RRBS method, but also we can see this with the like Horvath chip and with other tools, for example, the Transcriptomic. The effect is very strong. Probably one of the strongest we observe is a rejuvenation based on parabiosis. So now it's very clear, and you can find thousands of papers now on the clocks. The clocks do work. They are very robust, but there are still issues with them. And actually, I would say many issues, and some of them are shown here. So they may be trained for damage, for function loss, for diseases, for mortality, for rate of aging. It's unclear what exactly is the best way to train the clocks, and which clocks best represent aging out of this type of clocks. Also, they may be trained for the entire organism, for tissue, or for single cell. And that's the whole another issue, how to, for example, construct a clock at a single cell, how it represents, for example, a tissue or the entire organism. Also, clocks many clocks have kind of multiple processes contributing to them. So, for example, it may be aging, but also adaptation to aging, kind of response to the aging process. Also, aging and kind of development. Sometimes when clocks are built, for example, for the period when the two processes run together, so there may be a contribution of both processes. Also, clocks that capture transition from the young to the old state not necessarily work when we transition from older to younger state. And also changes may be stochastic and regulated. And also there are differences when we study decreased methylation with age and increased methylation. So there are all of these issues that are associated with the clocks, and we try and to address them one by one. And of course, this is ongoing work. There's no solution, and I'm not aware of anybody who has the solution, but I would stress that clocks are already very good, very robust, but we need to understand them much better. At least that's my thinking about it. And I'll show you a couple of examples of how we do it. So one is that we develop a first single cell clock, and here is the paper we published in about a year ago on this scalable probabilistic framework for epigenetic age prediction. Then just show you one example how it works. This is the mouse liver, the individual hepatocytes, 4 months old. You can see this, the age can be predicted quite well for various individual cells. This is hepatocytes from older mouse, and these are mouse embryonic fibroblasts, also individual cells. It can be predicted. So we study different cell types and tissues, and the clock works pretty well across tissues. We sometimes find that some cells, they age slower, like certain like stem cells, like muscle stem cells, and some cells like here, for example, a cell ages faster, so we think it's a senescent cell. And what model emerges from these studies is that even though the majority of cells, they age kind of in a coordinated manner, some cells age slower, like certain stem cells, and some cells age faster. And so in the end, when we compare to the bulk tissue, when we have like just a one kind of predicted age, actually the ages of different parts of the tissue and also different cells within the tissue may be slightly different. So this is a big challenge, how we address this in the future. How can we study in a very high throughput way kind of aging of individual cells? So a second challenge that I want to highlight is the difference in DNA methylation with age when we study increased methylation and decreased methylation. And here is a perspective we published a month ago with Brad Bernstein, and it deals with hypermethylation. And hypermethylation happens in cells when they divide, because the methylation state is imperfectly copied. like here is shown there's 1 cell becomes 2 cells when divide 4 cells and so on. And these are the methylated cytosines. You can see that they eventually may be lost. This hypermethylation state has various features. For example, it has features of senescence, activation of type 1 interferon genes, transposable elements, and these are some of the adaptive changes that protect against cancer, but that also contribute to aging. So, and this hypermethylation happens kind of across the genome. It's kind of in a more or less everywhere, kind of intergenic regions, within the genes and so on. It's quite different from the increased methylation, which is especially prominent in kind of regulated regions like the enhancers and promoters and associated with development. So that's also a difference. So another feature here is we study stochastic versus regulated changes. And just as an example, you can see that in the youthful state, methylation could be quite consistent, but then we may expect that some of the changes may be co-regulated. When one cytosine changes methylation, the other one would change in the same way. But then also could be stochastic changes, kind of random changes everywhere, as well as mixed changes. And when we study, we did observe, of course, we study here at a single cell level, we observe co-regulated changes in the mouse in this case, but also we observe also stochastic changes as well as mixed. So this is again a challenge, like to understand the contribution of these two processes. And the next challenge is a challenge associated with aging versus adaptive changes. And here I would like to illustrate this by this mid-journey image. So this is the same man in the younger state and older state. And let's compare what exactly happens during aging. So one feature we observe is that his hair becomes gray, and also there's loss of hair. And this is a feature of aging, right? So if you build clocks based on age-related changes, this particular feature may very well end up in the clock. However, these are neutral changes. They are kind of not causal. They accompany aging, but do not cause it, you see? There are some other changes we observe here, for example, the use of glasses, or like the use of hearing aid. Or if we can extend this concept, we could, for example, quantify the number of visits to doctors visits, or like the use of wheelchair, cane, and so on. So we could build a clock based on these features. But of course, they change with age, but they are adaptive changes. These are responses to aging, not really aging per se, right? And so in this case, in the case when we like fire all doctors, there will be no doctors, there will be no glasses, no hearing aid. Based on this clock, everybody will be young, which is of course not the case, right? So this tells that these adaptive changes, which similarly happen at the molecular level, they represent a response to aging, not aging itself. And therefore, when we build clocks like this, whether it's an epigenetic clock or transcriptomic clock, this component will be there. And if we happen to target these adaptive changes, we actually just remove a protection. We don't, target aging. We would observe artificially that this organism would be rejuvenated. So this is I think it's quite serious challenge, how we can do it, how we need to be addressed. And so we, Albert Ying in the lab, so he attempts to develop what we call causal epigenetic clocks. He uses Mendelian randomization and age-related changes to identify sites which are A, change with age, and B, they are either causal to aging or adaptive, protective against aging. And so these are the clock. It's a human blood clock based on all causal sites, and this clock is based on the aging sites. Yeah, so sites which kind of decrease lifespan, which contribute to aging. So we can build clock, and we also can build clock based on adaptive changes which protect against aging. And now let's apply these clocks to kind of real situation. And one example I show here is the example of reprogramming, cell reprogramming, when we know that we could convert by expressing Yamanaka factors somatic cells to embryonic state, like iPS cells. And the DEM age, we call this DEM age clock, kind of aging, damaging clock, actually shows rejuvenation as it should. Whereas the clock we call adapt age shows adaptive changes. Kind of there are random changes. It does not show rejuvenation. Just it gives an example that this separation, it's not a perfect separation, but it already tells us that we're probably in the right direction and try to deconvolute the age of the changes into various components. In this case, kind of aging changes, damaging versus adaptive changes. This is another example of the application of these clocks. So here we compare control and omega-3 fatty acid supplementation, and none of the existing clocks can capture the difference in this case, whereas adapt age and DEM age, they can capture. In fact, we observe the effect in the opposite direction. Even they both can quantify transition from young to old very similarly. So now, in addition to the clock, epigenetic clock, we also have transcriptomic clocks, and here is the study. This is actually clock is very good. We think it's one of the probably the best transcriptomic clock currently in the field. So this is based on more than 3,000 samples, and we incorporate both transition from young to old, as well as the study lifespan adjusted kind of changes. Here we incorporate the effects of interventions. For example, we sequenced most of the ITP interventions from the interventions testing program, NIE program, as well as other models that we developed. For example, as I said, parabiosis, mini interventions, and so on. The clock is very good. We currently apply it in many studies. There are many collaborations have been formed because there are so many RNA-seq data sets, and it also provides insights into the biology associated because of the availability of gene signatures. Just to show you one example of the use of the clock, here we study again reprogramming with Yamanaka factors, and you can see that this clock captures changes during reprogramming in the mouse, but also in the human. You can see difference in the timing of these events, and it's known that it takes longer time for human cells to be reprogrammed than the mouse cells. But again, it's just one example. So finally, I wanted to tell a little bit about the relationship between aging and development, at least tell about our thinking about this process. And just to start, I want to highlight a paper we published a few months ago, where we, in the mouse model, basically used rapamycin only during development. So we started with the newly born mice, and through the dams, we were giving rapamycin only for 45 days of life, and then did nothing. So, and this is the growth curve. You can see that this is the treatment window very early in life, and this is the control mouse, and these are treated mice. And you can see that actually we very quickly see growth retardation, both in males and females, and we and they never capture and never reach kind of the state of the controls, even when we stop rapamycin treatment. So what we find that these mice live longer, both well, actually, it's males is a much stronger effect than in the case of females, but it's very consistent effect. And I would just say that we published this study, but in parallel there was another study published from an Italian lab, Luca Tiberi. They also found the same effect. It's quite remarkable that two studies found this, and they also studied in flies, and they observed in flies, and they also collaborated with Leon Peshkin to study this in Daphnia. And again, we observed that treatment only during development, actually here in any dose, extends lifespan of these animals. So the effect is very reproducible. So it's interesting that when organisms can be targeted during development only, we already can target aging this way. So now to extend this studies on the development and aging, I wanted to introduce this concept of August Weismann on the separation of ageless germline and aging soma. And so, of course, this is a notion which is kind of well-known notion in the field, that germline does not age. It should not age in a sense that if the germline would age, then the next generation would be older than the previous one, and then the following is even older and older and would be quickly extinct. Of course, this does not happen. So, however, on the other hand, germline, these are live cells. They accumulate age-related changes and damage, for example. So, such as byproducts of metabolism or epimutations, mutations themselves, of course, as well, modifications in long-lived proteins. So, just from the conceptual standpoint, they are expected to age. Then we observe a clear contradiction that it should not age, but it ages. So, for new life to begin in the same young state, biological age somehow must be reduced somehow, somewhere. And so we have a hypothesis that germline may be rejuvenated in the offspring after conception, because we think it's at least initially that this is the best kind of place for natural rejuvenation. And when we applied our epigenetic clocks, and here is the study by Csaba Kiripesi from our lab, we indeed observed that the predicted age of the zygote, this is at the conception date, is quite low, but not zero. And then it reduces to a period where approximately 7, maybe 8 days post-fertilization, and it reaches the lowest age. We call this age ground zero. And then predicted biological age begins to increase. And we think that the aging process begins here, not here, but here, kind of in mid-embryogenesis. So when we applied this clock, and actually this is just one example, but in this paper we describe application of basically all kind of most robust clocks. They all show the same effect, so it's quite consistent effect. And then we also studied this at a single cell level using the single cell clock that we developed, and we also observed that changes in the period from 4.5 days embryonic days to 7.5 days. You can see at the single cell level also we observe a decrease. Of course, one may wonder whether this is because of the remodeling of the DNA methylome, and maybe it could be corresponding to the decrease in DNA methylation. But actually, when we study total methylation, we observe the opposite. So the lowest biological age at 7.5 days here in this data set corresponds to the highest methylation. So what we observe actually that, again, so methylation is highest at the lowest predicted epigenetic age. So basically what this means that for the effect of rejuvenation, we need first to demethylate the genome and then remethylate anew. And then this would result in the lowest biological age. It's not just the changes in DNA methylation. So so we also studied this a little bit more in detail, because at the single cell level, we can study different cell types during development. And we observed that, for example, in the epiblast, this is 4.5 days, 5.5, 6.5, 7.5 days, we observe a rejuvenation effect. Then we also see this in the primitive streak, endoderm, mesoderm, ectoderm. So they feels all three germ layers kind of they are rejuvenated, but interestingly, not in the case of extraembryonic tissues. Some of the extraembryonic tissues, they do not seem to be rejuvenated. So that's already, we still don't understand the mechanism, but we're actively studying this. So very interesting. So, but of course, one worry is that it's only described in our lab, so we really want other labs to verify this, to extend this study. And so currently we try to understand it through completely different orthogonal methods. And I already mentioned the transcriptomic clock. So we, of course, applied our transcriptomic clock to the embryonic rejuvenation models. And we also observed, this is in the mouse, that the predicted age of the zygote is again low but not zero, and it decreases during early embryogenesis with the lowest exactly at the same kind of ground zero state in the mouse, and then we observe an increased biological age based on the transcriptomic clock. And finally, we also extended this to the frog model, and here we study frogs because we could study thousands of embryos, and it's a very convenient model. For this, we had to develop our own epigenetic clocks, and we observed that now we're plotting here. 0 is the actually gastrulation, because this is the period where we think that corresponds to the lowest predicted biological age. We can see a decreased entropy, DNA entropy. We observe highest methylation at this point, and also predicted DNA methylation age also decreases. So here, approximately this is the minimum corresponding to gastrulation. So so far, mouse, human, frog models, different clocks, single cell clock, bulk clock, transcriptomic clock, they all show the same thing. So, but again, so it would be nice to extend it further. So interestingly, the developmental biologist Louis Wolpert once said, "It's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." And I just fully agree with that statement. They think that, but the reason it's important is because aging begins at that point. So this is our model. Again, so at the conception, age is low, is quite low, because germline in the previous generation ages very slowly, and then there is a rejuvenation. It's a first natural rejuvenation event, which can be captured by different clocks, and of course, it's very appealing to understand mechanisms of that rejuvenation event, and perhaps apply this in the future to somatic cells or other models. These are some of the papers we published so far on this topic. So where do we stand now? So this is our current thinking and about aging, development, and rejuvenation, which we learned actually a large extent due to the use of the epigenetic clocks. So we think that aging really represents accumulation of damage, and it increases with age. So it begins at ground zero. So in mouse, it would be probably around 7.5 or so embryonic days. In human, it would be maybe slightly after 2 weeks, between like immediately after 14 days or so. And it has no purpose. There's no benefit in having aging. On the other hand, development is quite a different thing. So it's a program. It's a regulated programmed event. It has a purpose to build a functional organism that would the highest fitness would compete with other organisms within the same species, with other species, and leave offspring. So it begins at conception, and then mostly ends around 20 years old in human, although it's also possible that it continues in some way. So for example, if we approximate this by differentiation events. So we think aging and development, there are two different processes. They go in parallel. It's not a process where it's aging is not something that continues after development. But I would say development in a way causes aging because it produces byproducts and damage in a broad sense. And then rejuvenation would then in this case would be kind of damage clearance, removing all these negative changes with age, which can be captured by clocks, but maybe not all clocks. So what about Weismann's immortal germline and aging soma? So the idea, of course, is great and still correct, but the addition would be that perhaps now it's clear that both soma and germline can age and can be rejuvenated. And of course, this is a very good news. I think that that means that aging can be targeted by targeting whether we could target germline or can target somatic cells. So, and finally, I wanted to summarize what I described today in my talk. So I discussed clocks, epigenetic clocks at the bulk level, single cell level, causal, which are damage-based, also clocks in the snake, morat, and frog, and also our new transcriptomic clock, still unpublished, we have particularly good clocks in the liver and also multi-tissue clocks. So I also highlighted a few challenges that still remain with the clocks, and I discussed this using the example of epigenetic clocks, but actually this applies to any clocks. And some of the challenges are this direction, because direction will be different for aging and rejuvenation. There are stochastic versus regulated changes, aging versus adaptation, aging versus development, differences in decreased and increased methylation, and challenges associated with building clocks at the organismal level, tissue level, cell level, and also trained kind of for different aspects of aging. So I briefly discussed absence, aging in the absence of increased mortality in the case of innate rats, where we think that this organism do age, even though their chance of dying does not change. Also, there are reversible changes in biological age upon severe stress. I showed you a few examples. And also there's an example of a paper published recently on aging during development, where we treated with a compound only during development, and also a couple of examples of rejuvenation based on heterochronic parabiosis, and also rejuvenation during development, culminating in ground zero state around the stage of gastrulation. But basically what we study, all of this is just to better understand aging and rejuvenation. So this is really our goal, and everything else is just tools for it. So finally, these are the people who contributed to this work. So I told you a little bit about the studies done by Bohan, Alex, Jesse, Albert, and Andrei, these 5 people, but also previous lab members, Alex, Anastasia, Chaba, and Sunny, they contributed as well. And of course, we are very grateful to really wonderful collaborators that we have. So most of the studies are done in collaboration with various labs listed here. And we're also very grateful to the sources of agencies and foundations that fund our research. So, and I would like to thank you again for your attention. Thank you.
Thanks, Vadim. Celia, do you want to start off the Q&A? It looks like actually Doe has a hand up as well, but there are some questions starting at the 7:57 a.m. mark, if you want to use that.
Yeah, maybe I can start with one here in the Q&A.
Oh, sure.
How? Sorry, is that okay?
Perfect.
Yeah.
Yep.
Okay. So how do aging clocks perform in the context of progeria? It's from an anonymous attendee.
Yeah, it's a very good question. They do not perform in some models of progeria, so it depends on the model. So I think again, so this needs to be also studied much better. We haven't really done a lot in this area, but we tested a few models, and sometimes they work, sometimes do not. Because progeria is not necessarily aging. Sometimes you could disrupt one particular process that can be easily fixed, and then the kind of you increase mortality, chance of dying on animals, and then but this may not be captured by the clocks because it does not necessarily mean aging. Yeah, in this case.
Okay, thank you. Doe, if you would like to ask.
Doe here. So that is a very excellent talk. So I know a lot of people have this fundamental question about the epigenetic clocks. So do you think these clocks are cause or consequence of aging? What are your thoughts? I'm curious.
Yeah, so in general, the easiest way to think about clocks is that they are biomarkers. They just correspond to aging. Aging happens and it's reflected in the biomarkers. But if you target biomarkers, nothing would happen. For example, if you have a clock composed of 50 CPG sites, and then you remove the CPG sites, it's not like aging would still continue in the same way. Or you could use other 50 sites to build a clock, which would be very, very similar to the original clock. But there are, of course, some sites which I try to describe this a little bit when I discussed causal clocks. So so in the contribution, there are some sites that do contribute. So we could capture those, and we call them causal clocks, because they actually literally shorten lifespan. In this case, I also see that with other health outcomes, negative health outcomes, or sort of a kind of protective, adaptive role. So they are also causal, just in the opposite way.
Thank you.
Thank you. Peter, see your hand raised.
Yeah. Hi, Vadim. Wonderful talk as always. So, some very provocative ideas there, which I don't necessarily disagree with, but obviously the idea that aging starts at gastrulation, or that naked mole rats, maybe age epigenetically but not demographically. Those are, very, very interesting ideas. I was wondering though whether you could, for example, with the mole rats, can you, I think this idea you have of developing a causal clock is obviously very powerful. So is there a way to build what you might say is a causal clock for the mole rat? And I guess if the mole rat is, as you say, really aging, then you should be able to generate a causal clock. On the other hand, if the mole rat is not aging, and what you have is just a bunch of methylation changes, which are completely passive and not really reflective of anything, then you wouldn't be able to build a causal clock. Is that something that you can try and do?
Well, theoretically, yes, we could try. Of course, in the current state of science, we cannot, because there is not enough data. So to build causal clock, we use at least in the human, it only can be done for humans so far, because we use this genetic variation kind of benefit from a huge kind of genetic studies. So this is not, there are no data for the naked mole rat, even for the mouse. It's very hard at this point to do it. But in general, this principle could be applied. Maybe in the future we can do it. But I think even a larger question is, actually it tells what is aging. So some people think aging is the increased chance of dying. You see? So, and these are all like our demographic models, many evolutionary models based on that kind of notion. So, but is it? So, because aging could be another view on aging is kind of damage accumulation or maybe functional decline. So what is actually aging? And apparently various investigators in the field we call aging completely different processes. You see, these are different processes. For example, in my mind, an organism can die for many reasons, for various reasons. It could be because of aging, could be something else. But in this case, naked mole rat does not show increased chance of dying, but in my mind, these animals, they still age, and this can be captured.
Yeah, just to be kind of devil's advocate on that. I mean, yes, there are many definitions of aging, but what I would say aging is not, it's not the passive, inconsequential change in a molecular marker, which has no functional consequence. Okay, and at least, I mean, it seems that a lot of the clock CPGs, I mean, that's what they are. They are, they are passive bystander molecular markers without functional consequence. So I think it's difficult to say that is aging when, over, for example, increased risk of morbidity and disease and death.
Well, there is a difference in opinion in the field. So again, what we call aging are different processes, and various people call aging different processes. So what is actually aging? and maybe there are different processes. For example, process of damage accumulation, process of functional decline, process of changes in fitness, and process of increased chance of dying. So these are different processes, and we all call it aging. But these are different processes, and of course they may be treated as different processes. And then we need to study, for example, which of these processes is best to be targeted in order to observe, whatever desired outcomes. Or maybe all of them, or maybe we need to call them by different names. We see.
Okay. So just as a kind of thought experiment then, I mean, I understand that you don't have the data to build a causal clock in the mole rat. Okay, but I'm wondering, what would you need to do that? You would need functional data across the lifespan of a mole rat to train that causal clock. Is that what you would need?
Well, we need population of hundreds of thousands of kind of individuals and with kind of very good genetics and very good genome methylation changes. So at least the method we used in the human case is just not applicable currently to the naked mole rat.
Right. Okay. Okay. Yeah. Very wonderful and provocative talk as always, Vadim. Thank you.
Thank you. Vera, could you like to ask?
Yes, well, great talk, Vadim. That will maybe be like a textbook in the future, your summary slide. So I just have a quick follow-up on Peter's question. I love this idea. I understand that this may not be possible, but what if, maybe using Steve Horvath's universal clock and looking at human sites that are also parts of the universal clock, then maybe you could get at that question.
Which question?
Peter's question about whether functional sites participate, for example, in the naked mole rat or in the universal clock for that matter.
It may be possible, yeah. We could translate, right? So what we find in human, maybe to other organisms, potentially it may be possible. So I mean, maybe we should try. Yeah, thank you.
Thank you. Morgan, I see also a hand right there. Thank you.
I also have a question about the causal clock, Vadim. Kind of two quick questions. So one is the causal versus I think you call it the adaptive clock. Are they enriched in either hypermethylation or hypomethylation? Do you find one that tends to be in one type or the other, or they're still kind of 50-50 like most clocks?
I would say 50-50. So yeah, there's no enrichment, but also this is the way we select, because we select for both kind of decreasing sites, decreasing methylation and increasing methylation. So maybe that's why.
And then the other thing is, so you use Mendelian randomization to identify, but what if we think that methylation changes are stochastic, then some of them may not have high heritability, and therefore it may be biased towards calling them non-causal because you don't have that kind of SNP.
Yeah. This can be viewed as the enrichment first step in the process, but of course, yeah, we don't claim to identify all causal sites, and yeah.
Thank you. I want to take one of the questions from the Q&A. So could you expect the age of the zygote to reflect the epigenetic age of the parents, or is it just higher than the ground zero? It's from an anonymous attendee.
Yeah, so it's interesting. We haven't really studied zygotes, not yet, from parents of different age. This would be very interesting. Yeah, but presumably yes, because there's clear that if we, for example, study a sperm, then the age of the sperm increases with the age of the organism. So presumably the oocytes will be the same, and then the zygote presumably also would show some differences. Yeah, that would be the prediction, but we haven't studied that yet.
Cecilia, do you want to take a few from the Q&A list? Yeah.
I'm just trying to, thank you.
No problem.
So from Sebastien Lemar, should we consider the result of DNA methylation reprogrammation as an age pattern, or is it a different axis of rejuvenation?
If I understand correctly, whether we just reprogramming, the question is about reprogramming, right?
I understand that is whether the DNA methylation reprogramming is an aging marker or a more for rejuvenation marker. So it is going towards aging or restoring the aging phenotype.
So when, okay, so if I understand correctly, the question is about cell reprogramming. So it's epigenetic reprogramming, but actually it's a cell reprogramming kind of captured by using clocks. Of course, DNA methylation changes during this period. I think it's a true rejuvenation when we start with somatic cells and end up with embryonic cells like iPS cells. Various clocks capture this, and these cells can be, for example, transplanted to blastocyst and end up in the organism, which is perfectly fine. So I think this is a rejuvenation event. I'm not sure I understand the question exactly. Yeah. Mm-hmm.
Okay, thank you. There is another one that from Gombo Liu. Say thank you very much for the great talk. Is there an exact time for the ground zero age in human or mouse?
Exact what?
Exact time.
Yes, timing.
For the ground timing.
So we have not defined it exactly. So we still don't know. So I say approximately. We say in the mouse maybe 7.5 days or so. In the human, as I said, approximately 14, maybe a little bit later. So in the human, because there are no data sets, scientists are allowed to work with embryos, human embryos up until 14 days. So there are essentially no data sets on the human side after 14 days, like between like 14 and 30 days, there are no data. So unfortunately cannot be quantified for that reason. So, but by comparing kind of mouse data and human data, it should correspond to that period approximately.
Okay, thank you. Let's see another one. So it's from an anonymous attendee. Say, thank you for the interesting talk. It looks like the epigenetic age takes place ahead of the biological age. And so do you think that the perturbation of the epigenetic aging markers could be enable rejuvenation? Or could the proliferation frequency and methylation level quantify the scale of aging?
It is possible that the targeting the epigenome, because it's such an important process, would change the biological age. But I should stress that aging, there's no like a main process in aging. So there's it contribution. It has contribution in my mind. It's my opinion from all different processes, and therefore it's kind of the epigenome is the reflection of it. It's a tool for us to understand better the aging process, but we cannot equate the epigenetic changes and aging, of course.
Thank you.